This podcast is supported by the Icahn School of Medicine at Mount Sinai, an international leader in research, education, and patient care. The medical and graduate school is part of the Mount Sinai Health System, one of the largest academic medical systems in New York City. Ranked among the top recipients of NIH funding, researchers at Mount Sinai have made breakthrough discoveries advancing the health of patients. Here, clinicians and scientists push the boundaries in cardiology, cancer, immunology, neuroscience, genomics, geriatrics, environmental medicine, and artificial intelligence. The Icahn School of Medicine at Mount Sinai, we find a way. This is the Science Podcast for May 28th, 2026. I'm Sarah Crespi. We've got a little bit of a theme going on this week. Keeping things alive or semi-alive when maybe they shouldn't be. First, it's whole brains in a tub. Freelancer Sarah Reardon talks about researchers keeping intact brains on life support to test drugs, and then the ethical considerations of these kinds of studies. After that, we're talking about keeping an amputated tube foot from a sea cucumber alive for 3 years in seawater. I talk with researcher Sarah Jobson about why her lab looked into this and what we can learn from such zombified limbs. Finally, this one's a little stretch on the theme, but it is the first in our 2026 book series. We're doing science biographies this year, and this one's on keeping Galileo's legacy alive. Books host Angela Saini talks with historian Anna Luna Post about her book on how fame shaped the scientific fortunes of Galileo. This week in science, freelance science journalist Sarah Reardon talks about using whole brains on a form of stripped-down life support to test drugs. And she heard about some of the ethical considerations that come along with these types of studies during a fellowship at MIT through the Knight Science Journalism program. We should probably hit right away on this very confusing aspect of this, as saying we're keeping the brain alive, we're keeping the tissue alive, but it's not a living brain. How do you talk about this?
Yeah, so that's something they've been really concerned about, is just making sure that this isn't some sort of fate worse than death where you have conscious brains continuing to exist in a vat.
The Matrix situation.
Yeah. Now, as the CEO told me, like the people who died, who donated their brains, they were brain dead for a reason. The people died, the brains no longer continue to function as full brains. They don't naturally have hardly any electrical activity, which is really what would be necessary for any sort of thoughts or consciousness or sensory processing, or like retaining memories, things like that. They also, just to be sure, they give them anesthesia. They give them too little glucose so that they're like kind of energetically starved and they can't fire, because they really just don't want to even run the chance. What they can do, though, is once they chop the brains up at the end into pieces that they can study in more detail, they are eventually going to be able to restore some of that electrical activity and really study any more brain functions that they weren't able to in these. But yeah, in terms of activity across the entire brain, there's no chance of that occurring, as they tell me.
It looks a little bit like a crock pot that has a brain in it.
Well, it reminded me of, I almost put it in the story, but I knew that it would get cut. It looks like a hotel ice bucket with like the little sticky lid.
It definitely does. Anything with food, they're going to say no.
Yeah. No, I didn't even try.
The story reminded me that when I was an undergrad, I worked at a lab, but I just worked with like frog cells and stuff. But to do the microscopy, I had to go to like a totally another building. And my friend was like, "Do you want to see something?" And it was in the neuroscience building. And there was just a bucket in the walk-in with a human head in it.
What?
Yeah. Hair, ears, everything.
Just as an undergrad, just walking by a head. That's wild.
Obviously, labs are usually locked and everything, but
Still,
let's go. Yeah, it was traumatizing for real.
Wow. Yeah, no, I would want a little more warning than something. I would have still wanted to see it, but that's...
I know. It just made me think, it's neuroscience. They need brains, but it was just weird.
Yeah.
But yeah, it really made me think of that when I was looking at the story and even the pictures. I was like, at least it doesn't have a face.
I took a close-up photo of it, but Bexark was kind of weird about it. They're like, we want to be respectful of our brains.
This story is interesting to me because we've been using human tissue. We've been using all this stuff for scientific research for a really long time. Preserved brains of recently deceased people, they have been really key to understanding neurobiology. If you think about all the lesion research, stuff like that. But having a semi-alive brain, something that is still living tissue and intact, I don't think I've come across that before. Is that kind of new?
Yeah, that's their whole innovation, to have an intact brain. Nobody's done this before at all, especially not at this scale. Endpoints did a story about them last summer, which I thought was really cool. And then one of their ethicists came and spoke in one of my classes just about how crazy it all was. They have 6 ethicists on their board.
Is that kind of the innovation here, really? Because keeping tissue alive in a sense that the cells are able to take in nutrients and continue to sustain some functions is not that unusual, right? Is the ethical part the innovation here, or is it the keeping the brain in a semi-alive condition?
That's the real innovation here, is just the ability to really test these things in real time, because the brain is the hardest organ to study with possible perception via placenta. Like in tumors, for instance, cancer biology, really being able to like take a sample of a person's own tumor, figure out exactly what's going on with it in the lab by testing drugs, has really revolutionized drug development in that area. But you can't do that in the brain. You can't just take a random chunk to see if is your drug actually getting into the cells or not. And you can't even see how the cells responding. We don't have a lot of like scans that will show you, or any sort of MRI or internal imaging that we would have for other organs for these kinds of drugs.
So why haven't people done this before?
Partly the technology. This paper that came out in 2019 with pig brains, they had set up this machine for the first time called BrainX, where they are able to flow this blood substitute through it, carrying oxygen, other nutrients into the brain, as well as filtering its waste out, everything the brain needs to be able to survive and to keep functioning, at least on a physiological level. They worked out a way to do that, found the brain was a lot more resilient than we thought it was, that it could actually stay alive outside, quote unquote, alive outside the body for a little bit longer. So they had that innovation. And that's happening with a lot of organs, I think, too. People are trying to keep them alive more for transplant purposes than research purposes. Yeah, it's kind of just took someone to do it and then build a company out of it.
So it's a brain from a recently dead person. Are they organ donors or?
They are, for all intents and purposes, organ donors. It's kind of amazing how many brains they've been able to get. Human brains are really notoriously difficult to get hold of in research. And these guys have partnered directly with organ procurement organizations that are these long established organizations that go and counsel people at the end of life and discuss what organ donation entails. And so it's not an organ that normally gets donated. So it's not like they're competing with others for it.
So you got to visit this facility where they're keeping these brains?
I visited a company called Bexorg in New Haven, Connecticut. They're right down the street from Yale.
Can you describe kind of what it looks like to have this brain on life support? I don't know what to call it.
Yeah, it is basically life support. They described it to me as like an ICU environment for the brain, like intensive care unit. It's this brand new space. This company's been growing really fast recently. They just moved into the space like a month ago. They've got these plexiglass cubicles all along their office floor, and each one inside has a cart with this brain in a bucket. And it has a lot of just tubes going in, flowing this blood substitute through a monitor. It's measuring hundreds of data points coming out of the brain, what its physiological functions are. If they sometimes could put a drug in, they could measure, is that drug being metabolized? Are we seeing these proteins in the blood that would indicate that? It's basically turned into sort of an assembly line where they have a neurosurgeon who takes the brain out initially back at the hospital. They send it up here to this company. They have one of their own surgeons who hooks it up to this machine for them, which seems like a pretty complicated process.
What kind of testing are they doing? You said something about they're giving the brains drugs.
Yeah, and that's the whole reason they're doing this. There might be some interesting, just basic neuroscience findings come out of this, but the real reason is that they want to figure out whether drugs are going to be working in the brain. It's so hard to test drugs in the brain and learn anything about what they're doing. So people use a lot of animal models, but animal brains don't function very well like human brains. It's hard to figure out like if the drug's even getting into the animal's brain. Same with things in a dish. You can't see if where a drug is entering the brain, for instance, or what its concentrations are in the frontal cortex versus the motor cortex. They're working with some other companies to try and develop Alzheimer's and Parkinson's and neurodegeneration drugs, especially. They're trying to understand how the brain's responding and how that might be diagnostic if they can find proteins that come out of the brain that has some disease that we haven't seen these before, then maybe we could find them in the blood of patients and use it as a diagnostic tool in the future.
There's no electrical signaling, as we've emphasized. They're, low glucose, they're anesthetized. Is that going to kind of impede some of this research, though? Because, what is a neuron if it's not firing and talking to its neighbors?
It may or may not. It depends on what kind of drug you're looking at and what kind of activity you're looking for. So say you have a brain with Alzheimer's that has these sticky amyloid plaques that destroy neurons during the disease. You could put your drug in and see, is it going after these plaques? Are the plaque size decreasing? None of that has anything to do with electrical activity, but it's still really useful information to have. But you're right. If you want to look at whether a drug is rewiring the brain during depression or something like that, you might need to study that in more detail. And so what they can do is look at these brain slices later with electrical activity to see if a drug might have a chance of causing seizures. In the meantime, though, we have been developing drugs for many decades before this company came along. So we have a lot of other assays we can use to study those sorts of things. So it's not going to be the only way that drugs are tested, but it's a useful one. And there's other things that can probably fill in a lot of the gaps, if not all.
The plan is to scale up to get more and more brains?
They think that they could have up to 1,600 per year, which is just wild. It's really useful to the drug companies working with them. The one that I mentioned in my story, Biohaven, they said that they've tested one drug in almost 100 brains. They can replicate 30 times, see what 30 different brains do, and then have 30 different controls. That's so much faster and easier than doing that 60 people.
Is there any regulation around keeping brains alive or whether or not electrical activity is allowed, either from the level of professional societies that scientists belong to or from the government?
Government, I'm actually not sure.
We just saw a lot of retirement of chimps from government research labs. There's been a real push to move research out of animals. And I feel like this is something that kind of aligns with that when you talk about the status of U.S. scientific regulation.
The company said that this has been really helpful for them. There has been this big push in the last couple of years for researchers, but basic research but mostly drug testing to move away from animals. And they don't really have a whole lot of better models. People are working on AI models. They're working on organs on chips. They're working on growing little miniature organs out of stem cells. And those have been super useful in their ways. And they let you test things like brain activity that you can't test in these Vexoric brains. But yeah, this is probably going to end up being a more direct model in terms of we know that the human immune cells react to this drug as opposed to a mouse's immune cells.
Yeah, that big gap between the lab animals and the human is pretty insurmountable. How are they going to make their money?
Yeah, they're going to make money by partnering, through a lot of things. Right now, they've had a lot of grants, just investor support. They're going to be partnering with companies to develop drugs. They've already had one partnership with a pharmaceutical giant called Biohaven, and there's a drug that Biohaven is now going to be testing for a neurodegenerative disease that was developed partly in these brains, in dozens of them.
And now they're moving it into people.
They're also developing a lot of things on their end. They're developing some of their own drugs because they just have this in-house testing network. They're using all of this data because they have this immense amount of data coming off of these brains, as well as all the medical history, genetics, activity that they might be able to look at from the brain slices. They're going to be looking at that in thousands of brains, which when you have a ton of data, it's perfect to train AI models, of course. So they're going to be creating this virtual brain that might even be able to test some new molecules in the virtual brain before you even ever go into a physical brain. That's not going to be a substitute for physical drug testing, but it might kind of cut some time off at the beginning and let companies test early or be kind of an early test bed for certain drugs.
Thanks, Sarah. It's been really interesting to talk about this.
Thank you for having me.
Sarah Reardon is a freelance science journalist based in Boston. You can find a link to the story we discussed at science.org/podcast.
No one goes to Hank's for his spreadsheets. They go for a darn good pizza. Lately, though, the shop's been quiet, so Hank decides to bring back the $1 slice. He asks Copilot in Microsoft Excel to look at his sales and costs and help him see if he can afford it. Copilot shows Hank where the money's going and which little extras make the dollar slice work. Now Hank has a line out the door. Hank makes the pizza, Copilot handles the spreadsheets. Learn more at m365copilot.com/work.
Did you know if your windows are bare, indoor temperatures can go up 20 degrees? Turn the temperature down with Blinds.com and get up to 50% off custom window treatments like solar roller shades and more during the Memorial Day mega sale. Whether you want to DIY it or have a pro handle everything, we've got you. Free samples, real design experts, and zero pressure. Just help when you need it. This is your last chance to shop up to 50% off site-wide during the Memorial Day mega sale at Blinds.com. Rules and restrictions apply.
Also on the site this week, I want to call attention to the news feature story by Jeff Mervis. This is about how the U.S. government has recently convicted multiple postdocs from China who are working here in U.S. labs for improper shipments of biological materials. Some see this as a replay of what was called the China Initiative in 2018, where the Trump administration was trying to exclude scientists from these international collaborations. Also out this week in Science, in the pages of the research section, we have a paper on homing pigeon navigation. It turns out that these birds are able to detect magnetic fields using macrophages in their livers. Next up, we have an interview with researcher Sarah Jobson on the regenerative powers of sea cucumbers and their lost limbs. We can keep some cell lines in a dish alive for an infinite number of divisions. But when it comes to, specific tissues or organs, there's a lot more that goes into it, and they don't survive long, even under culture conditions. This week in Science Advances, Sarah Jobson and colleagues wrote about taking a piece of tissue from a sea cucumber and getting it to survive in plain old seawater for multiple years. Here's our interview. What made you decide to look into whether or not when you cut something off a sea cucumber, it lives forever? Why did you even think of that question?
That is an excellent question. I would say that in the lab, the Mercier lab where we work, we are very curiosity driven. Oftentimes we follow what appears to us in the natural environment. So one of my colleagues actually noticed when we were working on this sea cucumber that amputated tissues weren't decaying at the rate that we would normally expect from animals generally. So we thought, that's weird. I wonder how long we could keep them alive. I wonder why they're doing this. And so we kind of just followed this keen observation, and it turned into something way more dramatic than we ever expected it to be.
So we should talk about your animal subject here. We've done stories before on reptiles, like a lizard losing its tail. The tail is this distractor. It helps it escape. But then the tail, it rots. Like you can't grow another lizard, but it also doesn't survive, right?
Exactly.
But you're working with sea cucumber. So is this something that loses limbs a lot or has limbs? I guess.
Yeah, this is great context because I think a lot of people aren't as familiar, obviously, with sea cucumbers. We don't see them as much. So this species is a sea cucumber called Solus fabrici. They do have, I guess, what we would call limbs. They have tube feet. They have tentacles.
Like a starfish has like little tube feet, right?
Yes, exactly. They're actually pretty closely related. We wanted to see what was happening with these tube feet because they are lost quite frequently. They are considered pretty vulnerable tissues. And so we thought, what is the regenerative potential in a limb that is being lost quite frequently in nature? They don't cast off their tissues, but they are lost in predator-prey interactions. And so it is somewhat similar, except that in the case of a lizard, it would be as if the tail was able to heal and survive and kind of wiggle away and live its own life.
You can see the adaptive advantage of being able to heal and then regenerate if needed. Some lizards do regenerate their tails, and these tube feet come back, and that's been a subject of study for a long time. Mm-hmm. But like turning to the tube feet and say, well, what are you going to do after you... And one thing you emphasize in this is the fact that you're not using culture medium, because like here we are in 2026 and you can culture cells and they can live a very long time, especially if they're stem cells or some more immortalized cell lines.
Yes, for sure.
But, they got to be in a little incubator with like special gases and special tissue medium and all that stuff. And that is not what is happening here with these sea cucumbers' feet.
Yes, exactly. So, as you mentioned, there's been a lot of research done onto immortal cell lines like HeLa cells, for example. And one of the reasons that they're so helpful is the fact that they're immortal. They can proliferate indefinitely, they can be used for long term studies. And the other is that they're very resilient, but they do need to be cultured in super specific, sterilized conditions. And so, these tissues, one, it's not just a singular cell line, it's a complex tissue, it's a group of cell lines.
The tube foot?
Yeah, the tube foot, yeah. It's a group of cells working together, and it is able to survive seemingly indefinitely, similar to HeLa cells. And as you mentioned, it is able to survive in a microbially diverse, completely unclean environment, basically just natural seawater. We've seen them surrounded by parasites, we've seen them covered in dirt, we've seen them in basically every unclean situation.
That's a lot different, even than organoids, which is where you take stem cells and then you influence them to differentiate. They do it with brains or hearts, and you're kind of getting distinct layers of cells, some functionality that kind of emulates what you're trying to get, like an actual organ. You get this organoid. But again, it is very, very specific culture, medium, conditions, and you're definitely not just throwing it into a cup of ocean water or
Yes,
whatever.
exactly. So it is, kind of as if you were to chop off your finger. It might be a little too gruesome, I don't know, but
No, that's
chop off
okay.
your finger and put it on your counter in like just a completely natural environment, and it were to heal and survive and just absorb everything it needs from the surrounding environment, because these tissues are able to maintain a metabolism. They absorb their nutrients from the surrounding seawater. They maintain an active immune system. They actively proliferate cells. They reorganize their tissues throughout the course of years. So they're maintaining some of these essential processes that are foundational to life, but they're not reproducing.
That's what I was going to say. It's alive. We're kind of skating the edge here because it's not growing. Like the tube foot is a mature tube foot. So it's not going through development. It does heal where it left the main body.
Yes. When it is amputated from the body or excised from the body, it has a big wound site. And within a few days, it's able to close that up, heal off the incision site, and get rid of any damaged tissue through apoptosis and use active cell division to proliferate new cells and heal off the original wound site.
Yeah. And you watch these over the course of multiple years in some cases. Do they reach kind of like an equilibrium where they're replacing cells, but not getting bigger or smaller?
At the stage of a few years, we weren't looking at their active cell proliferation anymore. We were more examining how their size was changing, what their external morphology looked like, things like that. And it does almost seem like they do reach a equilibrium of sorts. They become a lot more spherical than they were at the beginning, and they start to maintain a pretty similar appearance across years 1 through 3.
We should kind of get into the biological parallels that you can draw from this. So like we're at sea cucumber, which is way over here on the animal tree. And then with starfish, sea urchins, they're kind of all related over there. But then you got to walk pretty far to get to mammalian. What can we learn from something that is so far away from us that can do this? Because ultimately, this would be an ideal system for studying tissue and
100%.
low hygiene culture conditions.
Oh, true. Super resilient, pretty easy to culture. But as you say, they are invertebrates. So relatively, they are pretty far away from mammals. They're pretty far away from where a lot of our medical research is currently being conducted. However, in terms of the tree of life, sea cucumbers are echinoderms, which are a basal deuterostome. So that basically means that relative to all other invertebrates, they are actually pretty closely related to mammals in the tree of life. And there is a lot of genetic overlap. Their immune cells are very similar to mammalian immune cells. They do represent a very interesting possibility for biomedical research and something that we need to explore further. It's out of my scope of research, but I think that there is a lot of possibility for using them in these areas and seeing how these tissues respond to things like disease challenge, because we're looking at how this complex tissue, how these different cell lines are interacting with each other in kind of a closed unit.
They act like cultured cells in that they're like absorbing nutrients from the environment. They're not receiving it from a digestive process, which is,
No, yeah.
I think sea cucumbers have that.
They
eat and
food,
excrete.
Yeah, exactly.
And you also tried this with other sea cucumbers, is it echinoderms?
Yes, yeah, perfect.
But they didn't show this long-term stability.
No, they do have a very high regenerative capacity. Like they still survived in natural seawater a lot longer than what we would expect from most organisms, but most of them didn't survive longer than a couple of months. That again points to there's something unique about this sea cucumber, about these tissues. It's not just the regenerative capacity. There must be like a suite of processes that are working together to support what we lovingly call zombie life, because we're not really sure what it counts as.
One thing that came up when I was reading this was this idea that they're low senescent, so they don't kill off their cells, or the cells don't die through this aging process the way our cells do. Is that possibly a clue to why this is happening? Because I can see the advantage of healing from a wound, even a big wound, growing a new leg, but I can't really see the advantage of keeping that discarded leg alive.
That's probably one of our most exciting next step questions, is kind of diving into the adaptive advantage of this, because we can talk about exactly like you said, dramatic regeneration is an obvious benefit to the living body that lost the tissue. But for this little piece of amputated tissue that can't reproduce, what are the driving selective pressures that are maintaining regeneration? It might be a byproduct of evolutionary adaptations over many years, but there could be something else going on that we don't know yet. I think one thing that's really important to confirm going forward is whether or not there is any cellular senescence happening and what might be causing this. But I think confirming for sure, from what we can see, they look to be immortal to us, but confirming that they are actually immortal.
3 years is a nice long time.
Yeah.
But we definitely have cell lines that have gone decades.
Exactly. There's so much more to be done.
We've touched on the idea of it being like a research model. How do you envision that being like a useful lab tool?
I think that being able to study tissues like the ones taken from Solus fabrici is really important, because there's still so much we don't know about regenerative biology, and sea cucumbers demonstrate incredibly high regenerative capacity. And so they've been kind of highlighted in previous research as an ideal model organism. But this species and these tissues kind of take it to a next level. And then there's also a ton of other interdisciplinary questions. It can go into medical sciences, looking at disease challenge. It can look at telomeres and cellular senescence. Also, it can give us a better look into ocean health as well.
Sarah, thanks so much for talking with me.
Yeah, it's been my pleasure.
Sarah Jobson will be receiving her PhD next month from the Memorial University of Newfoundland. You can find a link to the Science Advances paper we discussed at science.org/podcast. Stay tuned for the first in our 6-part monthly series on scientific biographies. Host Angela Saini talks with historian Annaluna Post about her recent book exploring how fame shaped the scientific fortunes of Galileo.
Hi there, I'm Angela Saini. Every month until October, I'll be interviewing the author of a scientific biography. Today's the very first book on our list, Galileo's Fame, Science, Credibility and Memory in the 17th Century by Annaluna Post, who's a historian at Leiden University in the Netherlands. This is not a typical biography. As well as exploring the life of Galileo Galilei, it focuses on how he became a household name. As Post explains, this didn't happen automatically. It was, she writes, the result of a long and intense fight over his fame that involved Italian and European artists, poets, courtiers, mathematicians, astronomers, philosophers, ambassadors, diplomats, noblemen, friars, priests, and cardinals. Just like modern-day celebrities, Galileo seems to have had his own PR machine. He also had his haters. Annaluna, what made you want to approach Galileo's story from this particular direction?
I think I've always been interested in the topic of celebrity and fame since I was very young, growing up in the 90s, 2000s. And then I never realized you could approach it as an academic topic. And I was introduced to it during my master's studies in Utrecht. I realized it was such a fascinating way of exploring the early modern period, but especially the world of learning. And then during my final stretch, I took a course in Rome on science and religion. And of course, Galileo popped up everywhere. And I decided he would make such a fascinating case study to pursue a PhD on. So that's what I did.
In your book, it is structured like a traditional biography in as much as we go through Galileo's life chronologically. So just starting at the beginning, then, what was he like in his early years when he was just an aspiring mathematician trying to get a job?
So Galileo was a bit of he couldn't really find his footing immediately in the university system of his age. He studied medicine, was pushed to do so by his father, who was a lutenist, who played music, and wanted his son to have a career that offered a bit more security. So he went into medicine, but he really didn't like it. And then he started to pursue mathematics instead. Galileo was the lower nobility in Florence, so he had quite a cultural network of important people around him. And I think he partied a lot, to be honest, with them. He had a really good youth and he had a good first, let's say, 25 years. But he also struggled after completing his education to find a position at the university. And he finally did so through those cultural networks that I just referred to. He started working in the University of Padova, which was one of the most important universities in Italy at the time. And then I think for him, something started to click, but it only really happened during his 40s. So Galileo really had his breakthrough moment in his 40s as a scholar, which is, I think, a fascinating piece of information for
and reassuring.
Exactly.
And just like with modern day scientists, you have to establish your credibility. You have to align yourself with certain networks and institutions. One of the factors I was so fascinated to learn in your book, shaping his credibility in those early years, was the fact that he came from Florence. Why did that make a difference?
So it was really important because Florence was known as a place of learning and of high culture. So it was the place where people like Petrarch, Machiavelli, Dante came from. That was a really important signifier of cultural status and wealth that Galileo built on and was able to build on. But what was even more important was that after Galileo started working with the telescope, he was doing so in Padua and in Venice, and he was really able to sort of leverage his work with the telescope in Padua into a more permanent position there at the university. But Galileo was hoping to go to Florence and to work at the court in Florence, where the Medici family ruled, because he thought it would allow him to focus more on his research and on writing books rather than on teaching, which is, again, I think, something that many academics nowadays will recognize. So he was really hoping to make the move to Florence, and eventually he did so. But then to people in Padua, that signaled his unreliability because he had started his work there, he had accepted a position there, and then he made this move and he sort of confirmed the already existing stereotype that Florentines were not just cultured, but they were also deceitful. And those sort of stereotypes, they play a role in the early modern world of learning. And I think that's really fascinating.
Yeah, it was really interesting to see how almost like racial stereotypes or regional stereotypes about people really affected how they were perceived at the time. Another thing that impacted his reputation then was at least one accusation of plagiarism. What was behind this?
So Galileo was not someone who was a very productive academic in the sense that he published a lot, especially in those first 40 years. But he did start working on this instrument, a geometrical compass, he called it, in Padova. And he would offer private tuition to students in his house to work on the instrument. And then he sold them a manual that he'd written. One of the students in Padova accused him of having plagiarized that instrument from another scholar. And the student, Baldassare Capra, wrote a manual in Latin to say that Galileo had plagiarized it. And Galileo, of course, felt that he needed to defend himself against these claims. And so Galileo turned to the board of the university, basically, to ask them to intervene, because this was, of course, a conflict between a student and a professor. And they organized sort of a scholarly trial in which they even asked the student, Capra, to perform certain mathematical calculations. And Capra didn't want to go along with it, but at the end it was exposed that he did not know how to work the instrument or that he couldn't have written the manual himself. So Galileo's reputation, was saved, but those accusations tend to linger.
Everything changed for Galileo in 1610. That was the kind of big breakthrough moment for him when he published his observations of Jupiter's moons, proving that the Earth was not the only body in the universe with its own satellites. And this quickly made him one of the most famous people in Europe. What was that like for him?
I think it was exciting, but it was also really quite difficult and challenging. The work really caused a stir, and the work sold out very quickly. People started writing him letters and asking for copies of his book, but also of the instrument, because Galileo's instrument was of a higher quality than many other instruments. And so people that did have a telescope could not necessarily make the same observations as Galileo, so they would accuse him of making things up. But he really made the conscious decision to send his best instruments to people that were of high aristocratic standing rather than of high scholarly standing. So that was another complicating factor. So people all over Europe start writing each other to ask what they have heard about Galileo's discovery, what they know about his character. And for Galileo, it's a frenzied period in which he's really trying to make the most out of the momentum that he's gotten, but also defend himself against these claims of plagiarisms, against these accusations that he's just made things up. He's trying to give demonstrations, but sometimes, the instrument, it's really tricky to work with. So sometimes people just don't have the eye quality to make the same observations. So it's really an interesting period, but it's also really difficult for Galileo.
And also around that time, there were various groups interested in promoting Galileo, including his patrons, of course, the powerful Medici family. What was in it for them?
In part, it's about prestige. For the Medici family, it's really about being the first family that has heavenly bodies, celestial bodies named after you. So that was really important. Around the Medici family, there's a group of poets who work at court, who are sort of tasked, or at least what they do is praise the Medici family as well as they can. So Galileo's work gives them another way of doing this. But for them, sometimes writing about Galileo's discoveries is also a way of playing nice with the cultural elite and gaining access to the literary academies in Florence. Then we have a group working in Rome, mostly, the Accademia dei Lincei, they're called, the Academy of the Links. And that's a really fascinating group because they work on new scholarship and they have this idea that scholarship needs to be based on observation. And so for them, Galileo's work is sort of an example of the success that can be obtained with this method. So they start really rallying around Galileo as well. So there's these different sort of things. It's about prestige. It's about appreciation for something truly remarkable, but it's also about those things intersecting.
And again, all very relatable, even though it happened such a long time ago.
Yeah.
And at the same time, of course, fame has its dark side. In 1633, we know that Galileo was put on trial for heresy for teaching that the Earth was not the center of the universe, which was the biblical view at that time. In your book, you suggest that it was possibly professional jealousy that helped to precipitate this, that Galileo's contemporaries thought he had got too big for his boots. That was the impression I got when I was reading, and ended up denouncing him then to the Inquisition in Rome.
So Galileo is appointed by the Medici family a full professor for the rest of his life without the obligation to teach. But still, his colleagues at the University of Pisa feel that he is a threat to the stability of the curriculum, which is mostly Aristotelian based, so which places the Earth firmly at the center of the universe. So they will attack Galileo because they feel he is threatening their students' right way of study, and that he's tempting them to stray from the Aristotelian path. Then there's a different group of opponents. They're Dominican preachers and friars in Florence, and they object to scholars making claims that go against the Bible. And those things, the scholarly world and the religious world, are very much intertwined. The danger for Galileo lies in the fact that they have access to channels that he cannot access himself and where he cannot really defend himself. He doesn't know what the exact allegations against him are. And those allegations are not just that Galileo is becoming too famous or is saying things that aren't true, but especially that people are presenting themselves as Galileisti, so as followers of Galileo, and also go around in Florence, in the streets, showing what people in the church consider to be untruths. That's the real danger for the Catholic Church at the time, that Galileo seems to be starting this movement that has the potential to disrupt authority and political order at the same time. Galileo is given a warning that he shouldn't teach heliocentrism anymore, but the wording of that order is quite vague. And so Galileo at some point feels that he does have the leeway to explore the topic hypothetically. But in a book he publishes in 1632, it's quite clear where he actually stands on the subject. And he also makes a very grave mistake, which is to put the favorite argument of the Pope, Urban VIII. The Pope essentially says, well, we cannot know for sure whether heliocentrism is true. And he has a favorite argument for that. And then Galileo puts that argument into the mouth of the stupidest of his three interlocutors in the dialogue.
That doesn't go down well with the
That
Pope.
doesn't go down well. No, of course not. No, it's an insult.
Yeah.
That's a mistake that Galileo made.
You do mention in the book that this charge of heresy by the Catholic Church was meant to destroy a person's reputation, not just in life, but also after death. And I do wonder, how was Galileo then remembered after he died in 1642? Of course, now he is held up as one of the greats of modern science, but how was it immediately in that time?
Galileo's contemporaries really try to do everything they can to make sure that aspects of his memory are maintained that the church wants to see gone. So essentially, the church wants him to be remembered in a different light. They don't want him to be forgotten, but they want him to be remembered as a bad scholar, but a good penitent, someone who essentially eventually listened to the Catholic Church and their orders. But his contemporaries, his supporters, try in different ways, openly, less openly, to defend him. In the end, they succeed in portraying him not just as a successful scholar, but also as someone who suffered for science. And that's a really important feature of his long-term memory, and something that is maintained until now, I think. Galileo is this martyr of science, which really brings together these ideas, again, of science and religion, and that also allow people nowadays to use Galileo for various ends. So I have encountered Galileo in many different settings, one of the most striking of which was during the marathon in Padova, where there was this portrait of Galileo next to the route to say, well, essentially say, keep going. You can overcome all difficulty, just like Galileo did, which is a very fascinating use of the image of Galileo, but he's really everywhere nowadays.
Yeah, he absolutely is. I did wonder, just finally, while reading your book, what reflections you might have now on modern day scientific celebrity, especially in the age of social media. What lessons or warnings are there in Galileo's life for us today?
I think it's really important to realize that celebrity and fame are nowadays not considered to be as dangerous as they were in Galileo's time. So some of the associations with vanity and with pride have disappeared, and yet some of them also linger. And I think that some of the mechanisms that make fame into a really ambiguous but important asset in the early modern period, like emerging capitalism, commercial pressure, and mass media, those mechanisms have only established themselves more firmly, which means that scholarly fame is more attractive to various parties as well. And that means that I think as a scholar, you need to be very aware and cautious about who you align yourself with, because science can be politicized to such an extent. And I think that's something that we can really take away from Galileo's case. But what the case also shows is that Galileo tried to exert influence over the way that he was seen by contemporaries and remembered. But his case also shows that it's very difficult as an individual to do that, and that celebrity and fame are really things that are made by groups. So in the end, we only have limited control over our own persona.
That's a good thing to remember. Anna Luna Post, thank you so much.
Thank you, Angela.
Next month, I'll be interviewing Georgina Ferry about her biography of the Nobel Prize-winning chemist Dorothy Hodgkin. See you then.
And that concludes this edition of the Science Podcast. If you have any comments or suggestions, write to us at sciencepodcast@aaas.org. To find us on podcasting apps, search for Science Magazine. And to find us on YouTube, search for Science Magazine Podcast. Or you can listen on our website, science.org/podcast. This show was edited by me, Sarah Crespi, and Kevin McLean. We had production help from Podigy. Our music is by Jeffrey Cook and Winkoi Wen. On behalf of Science and its publisher, AAAS, thanks for joining us.